Hello everyone. Today I will talk about the effects of duration time of heat treatment on amino acid digestibility in enzyme treat soybean meal fed to willing pigs. Let's start with the introduction. Soybeans are one of the most important agricultural crops around the world. The annual production is around 389,000 million tons per year, and the main products are oil and soybean meal. Around 93% of these soybean meal are used in animal production. This is due to the nutrient composition of soybean meal. Soybean meals contain around 70% of total dietary fiber, another 15% of other carbohydrates. However, the main nutrient in soybean meal is crude protein, which is around 47.73%. Also, soybean meal is considered one of the plant crops with high concentration of lysine, which around 2.97%. However, soybean meal contains anti-nutritional factors such as trypsin inhibitors and oligosaccharides and other antigen proteins which may reduce the digestibility of nutrients. The tolerance values in willing pigs for trypsin inhibitors in diets should be lower than 4.7 milligrams per gram. And for oligosaccharides, it should be lower than 1%. There are some treatments to reduce or maintain reduce anti-nutritional factors such as heat treatment, which helps to reduce trypsin inhibitors, lectins, and some antigens, and enzyme-treated soybean meal, which helps to reduce trypsin inhibitors, leptin, oligosaccharides, phytate, and antigens. Let's talk about a brief description of the production of soybean meal and enzyme-treated soybean meal. This process starts with soybeans, and after a crushing process, which is a oil extraction, we obtain oil and the fat soybean meal. After we have the fat soybean meal, there are an additional drying step to reduce anti-nutritional factors and excess of hexane if hexane was used. Once the soybean meal production is done, it is necessary to rehydrate the soybean meal and inoculate it with enzymes. After the enzymatic treatment is done, there is an additional drying step to remove the excess of water. In these two steps, there is a risk of heat damage. One of the main reactions in heat damage is the Maillard reaction. Maillard reaction is the chemical reaction between amino groups and reducing sugars under certain conditions, such as temperature, humidity, pressure, and time. As long as the process, if there are amino acids and sugars, the process will continue. It can happen with all free amino acids. However, in proteins, the amino acids most affected are lysine and arginine. But why? This is the chemical structure of lysine. It is also known as a reactive lysine and is the lysine that the animal uses for protein synthesis. Lysine has two amino groups. This is the first amino group that binds another amino acid to form protein, and this is the epsilon amino group, which is free and can bind to a reducing sugar. This new structure is known as unreactive lysine, and this unreactive lysine cannot be used by the animal for protein synthesis. However, there are no studies that shows the effect of heat damage on enzyme-treated soybean meal. Therefore, the objective of this study is to test the hypothesis that overheating will reduce the concentration of reactive lysine and the standardized ileal digestibility of lysine in enzyme-treated soybean meal fed to willing pigs. Let's move to materials and methods. One source of soybean meal was divided into batches. One batch was used without pure treatment, and the other batch was used to produce enzyme-treated soybean meal. The enzyme-treated soybean meal was divided in 4 different batches, and they were autoclaved at 120 degrees Celsius. For this experiment, we formulate 6 diets. Diet 1 contained 40% of soybean meal and 30% of cornstarch. Diet 2, 3, 4, and 5 contained 35% of enzyme-treated soybean meal and 35% of cornstarch. The difference between these diets is the enzyme-treated soybean meal used. For diet 2, we use non-heat damage enzyme-treated soybean meal. For diet 3, we use 30 minutes heat damage enzyme-treated soybean meal. For diet 4, we use 45 minutes heat damage enzyme-treated soybean meal. And for diet 5, we use 120 minutes heat damage enzyme-treated soybean meal. Also, we formulate a nitrogen-free diet to determine basal endogenous losses and to calculate the standardized ileal digestibility. Each diet also contains 0.4% of chromium oxide as an indigestible marker. For this experiment, we used 12 cannulate pigs with an initial body weight of 11.38 kilograms. The experimental design consists of a 6 by 6 replicate Latin square with 6 diets and 6 periods. Each period involved of a total of 7 days, with 5 adaptation days and 2 collection days. The collection period was 9 hours per day. Let's move to the results. In this slide, I want to show the effects of enzyme treatment on nutrients. In the y-axis, we have the concentration of the carbohydrates in percentage, and in the x-axis, we have each nutrient. In blue bar, we have soybean meal, and in the orange bar, we have enzyme-treated soybean meal. In this graph, we can observe that after enzymatic treatment, there were a reduction of stachyose, raffinose, and sucrose. However, there is galactose in enzyme-treated soybean meal, and it is possible that during the enzymatic reaction, the oligosaccharides are breaking down and galactose units are free. In this graph, we can observe that there is an increased concentration of dry matter due to the drying steps after the enzymatic treatment, and because of the reduction of sucrose and oligosaccharides, ash and crude protein concentration increased. Now, I want to show the effects of heat on nutrients. In the y-axis, we have the concentration of the carbohydrates in percentage, and in the x-axis, we have non-autoclave and autoclave enzyme-treated soybean meal. In the dark blue bar, we have stachyose. In orange bar, we have raffinose. In the green bar, galactose. And in the light blue bar, sucrose. In this graph, we can observe the concentration of carbohydrates is reduced when the time in the autoclave increases. Also, we can observe the effects of heat on amino acids. We choose the 5 most affected amino acids as in the previous slide. On the y-axis, we have the concentration of amino acids in percentage, and on the x-axis, we have non-autoclave and autoclave enzyme-treated soybean meal. In the dark blue bar, we have non-autoclave enzyme-treated soybean meal. In the orange bar, 30 minutes autoclave enzyme-treated soybean meal. In the green bar, 45 minutes autoclave enzyme-treated soybean meal. And in the light blue bar, we have 120 minutes autoclave enzyme-treated soybean meal. All the amino acids decrease when the time in the autoclave increases. Arginine and lysine decreases due to Maillard reaction. Reactive lysine also has a similar behavior to lysine. However, reactive lysine is always lower than total lysine. Cysteine is highly sensitive to heat because its sulfhydryl group forms cross links or oxidized, reducing its availability. And tryptophan can degrade through oxidation under high temperatures. This slide was set up as the previous one. On y-axis, we have the digestibility of amino acids in percentage, and on the x-axis, we have non-autoclave and autoclave enzyme-treated soybean meal. In the dark blue bar, we have non-autoclave enzyme-treated soybean meal. In the orange bar, 30 minutes autoclave enzyme-treated soybean meal. In the green bar, we have 45 minutes autoclave soybean meal, and in the light blue bar, we have 120 minutes autoclave enzyme-treated soybean meal. For the standardized ileal digestibility of all amino acids, all the amino acids have a linear reduction with a probability lower than 0.01. The amino acids with the stars have a quadratic reduction with a probability lower than 0.05, and the rest of the amino acids tend to be reduced with a probability between 0.05 and 0.1 due to the increased time in the autoclave. With this, we can conclude that the enzymatic treatment reduces the concentration of oligosaccharides and sucrose and increases the concentration of dry matter, ash, crude protein, and amino acids. As the enzyme-treated soybean meal has reduced oligosaccharides, it can be incorporated in diets for weanling. Also, heat damage reduces digestibility of amino acids and the concentration of lysine and reactive lysine. With this, I would like to thank all the STAIN Lab members and thank you for listening us. And please follow us on all our social media.